The central dogma of molecular biology refers to the processes of replication, transcription, and translation. Replication is the process by which double-stranded DNA is carefully unzipped, each nucleotide pairing with a complementary nucleotide, producing 2 strands of DNA from each one. Transcription is the process by which DNA is transcribed into the language of mRNA to code for proteins rather than store genetic information. And translation is the process by which that single strand of mRNA is translated from a string of nucleotides to a string of amino acids, which then refold and may undergo further processing to become any of the plethora of functional proteins needed for life. Francis Crick, one of the people accredited with determining the 3D structure of DNA, referred to the final translation step as the adapter hypothesis. To understand what he means by this, let's dive further into the mechanisms behind how translation occurs. This is our single strand of mRNA, or messenger RNA, with a variety of nucleotides arranged in 3 nucleotide sequences called codons. Each codon codes for a specific amino acid. The amino acid is carried to the mRNA by a different form of RNA called tRNA. Each tRNA has an anticodon, which is a sequence of nucleotides on its base that are complementary to the codon on the mRNA. This allows them to pair with each other. Each tRNA is charged with a specific amino acid that corresponds with its anticodon, put in place by an enzyme called aminoacyl synthetase. Crick was describing the tRNA matching each codon on the mRNA with the specific amino acid needed when he called it the adapter hypothesis. A specific sequence of nucleotides upstream from where translation is set to occur matches with a sequence inside the ribosome, allowing the ribosome to bind to the mRNA. The ribosome is a protein which houses the mechanism by which the amino acids carried by the tRNA are attached together. Once all parts of the ribosome are in place, tRNAs will begin to pair with their respective codons, moving through the ribosome and depositing their amino acids next in the string before being released to find an aminoacyl synthetase enzyme to charge it again. The cycle repeats for as many amino acids are necessary for the protein to be completely assembled. Let's model this process using real-life items to help us understand. In this example, the laptop is a specific codon in an mRNA sequence, and the cell phone is the amino acid. The phone and the laptop have no compatibility with each other, so how can we connect them? We use an adapter cable that has a fitting on either end that matches with both the laptop and the cell phone. This cable represents the tRNA by bridging the 2 disparate entities. My hand acts as the ribosome, connecting the adapter to the phone and the laptop. A few important distinctions to make. First, during translation, the ribosome first connects to the mRNA, not to the tRNA, as shown in my example by my hand carrying the cable to the laptop to plug it in. Second, the ribosome holds 3 tRNA molecules at different stages of the translation process at a time, unless you're at the very beginning or the very end of translation. Third, in my example, there is no analogy for the process by which the amino acids form a long chain. If we imagine a series of multitudes of laptops side by side, all connecting to different cell phones and sequenced via adapter cables, and if we somehow imagine that all those cell phones then sequentially attach to each other, we may have a more thorough analogy. Problems could arise throughout this process, however. An example would be if the aminoacyl synthetase attaches the incorrect amino acid to the tRNA, so that the incorrect amino acid, or in this case, a video game controller instead of a phone, will attach to the strand of amino acids. This will lead to improper folding and will likely result in a non-functional product, which could result in a number of side effects, including cell death.